Good morning. I have the pleasure to introduce my colleague, Professor Nesrine Salah Eldin. Is a professor and head of Pharmacology and Toxicology Department, Faculty of Pharmacy at Cairo University. In addition, she is the director of the International Relation Bureau in the faculty. Her research interest is focused on the molecular mechanism underlying the signaling pathway of many neurodegenerative diseases, with in particular experience with animal model. The seminar will focus on drug repurposing, and repositioning for Alzheimer's disease. Please, Nesrine, thank you for your seminar.
Thank you. Good afternoon. You hear me? As Professor Paula said, I'm Nesrine Sayed, professor and head of the Pharmacology and Toxicology Department in the Faculty of Pharmacy, Cairo University. At first, I would like to thank very much Professor Paula and the Faculty of Medicine for their kind invitation to join you today in our seminar. Briefly, at first, I would like to give a briefing about our university. Cairo University include many faculties. We have the faculties involved in life sciences and social sciences. Usually there are 26 faculties, and our faculty has 9 departments. And usually we work as interdisciplinary with other departments in our faculty. 15 years ago, we started in our pharmacology lab working on drug repurposing and repositioning in neurodegenerative diseases in general, but we are focused recently on Alzheimer's disease and to less extent on Parkinson's disease, which are both neurodegenerative disorders. Usually we work on in our lab on animal models. They are not transgenic models, but they are models that we select to mimic some pathology and cognitive functions of Alzheimer's disease. Of course, we cannot prove in our model all the aspects and the pathology of Alzheimer's disease, and usually we focus at first on the drug and on the mechanistic pathway, and then we select our model. Then we implement the research and the project of working on certain drugs and of course certain phytochemicals in the management of certain aspects of Alzheimer's disease. So I wanted to give you a briefing in our work, but at first I just mentioned some introduction of Alzheimer's disease. You know, of course, that Alzheimer's disease is a progressive neurodegenerative disorder. Progressive is very important as it occurs in many stages, starting from early stage, then middle, then severe, and finally death. So for this reason, it is called the progressive. And it is neurodegenerative disorder, which means that we are focused on neuronal degeneration, which results into loss of certain areas of the brain, and of course, certain skills and the cognitive function of the animal. It is usually a condition of old age and has a long preclinical period of approximately 20 years. So we can progress from early stage up to the third or severe stage in the period of 20 years. So our aim all over the world is to diagnose as early as possible the symptoms of Alzheimer's disease to try to stop the progression of the disease. And as you all know, that there is no treatment actually of Alzheimer's disease except of some monoclonal antibodies which are emerged in the market, I think 20 years ago. But of course, there could happen some side effects. So it is better to stop the progression rather than to try to treat, because once there is neuronal degeneration, we cannot regenerate these areas again. Okay. Then talking about the hallmarks of Alzheimer's disease, it is marked by the accumulation of amyloid beta plaques found in extracellular spaces. and the blood vessels wall, and the aggregation of neurofibrillary tangles or tau protein in these neurofibrillary tangles. Usually, as you see in the difference between normal condition and the Alzheimer condition, we find that here there is intact neurons, and the connection between every two neurons is intact. And it is shown in the hemisphere with the brain of healthy individuals. When we see, on the other hand, on the other part of the picture, the Alzheimer disease, we found two important pathologies which are present in Alzheimer disease. The first one is the deposition of the amyloid beta plaques, which is a very important pathology seen in patients with Alzheimer disease. And the second one is the formation of neurofibrillary tangles, which results into loss of neurotransmitters between two neurons, and of course, loss of nutrition and chemical messengers between these two neurons. So the net effect is, as you see, there is a neuronal degeneration, loss of brain structures, and of course, a loss of neurotransmission and the appearance of the symptoms of cognitive dysfunction. Of course, when there is a loss of certain areas of the brain, like the hippocampus, it is the main area concerned with the memory. And for this reason, that the first symptom that we notice in Alzheimer disease is loss of memory, because the first structure we find that suffer from the neurodegeneration is the hippocampus. After that, there is another structures which are lost, concerned with the thinking ability, the language, the calculation, and many other features. So in short, when we summarize the pathology of Alzheimer disease, it includes the formation of amyloid beta plaques, which is due to overactivity of beta and gamma secretases enzymes, and then it will result into formation of amyloid beta oligomers, and then aggregation to the amyloid beta plaques. On the right side, we find the tau hyperphosphorylation, which result into formation of neurofibrillary tangles, and then result into neurofibrillary degradation. Also, there is an important impact of neuroinflammation during the Alzheimer disease pathology, which include neuronal apoptosis, hypersensitive microglia, and blood-brain barrier disruption. All of them will result into neurodegeneration, which of course result into cognitive impairment, memory loss, and mitochondrial impairment and synaptic dysfunction. All of these pathology, including amyloid beta plaques, neurofibrillary tangles, and neuronal degeneration, all of them will result into the symptoms and pathology of Alzheimer's disease. What about the prevalence? The prevalence of Alzheimer's disease is growing all over the world. And we have approximately 50 million people worldwide are living now with Alzheimer's disease or other types of dementia, with numbers expected to reach 82 million by 2030 and 152 million by 2050. So we expect that this number will triple by 2050. So we have a still growing population of Alzheimer's disease. And of course, we can notice that there is another number of people who are not diagnosed. So we have many problems all over the world of the late diagnosis of Alzheimer's disease rather than early diagnosis. And as I mentioned, that early diagnosis will allow us to target and to start early treatment or early management to prevent a progression to next stages of Alzheimer's disease. Concerning also the prevalence in the United States, for example, more than 60 million people are living with Alzheimer's disease, with projections indicating the number could reach nearly 13 million by 2050. What is the impact of Alzheimer's disease growing prevalence among the population on the economical burden, healthcare system, and the quality of life? We will start by the economic burden, which is very important impact of Alzheimer's disease. Alzheimer and the other dementias cost the United States, for example, $352 billion in 2021, including 2039 billion in Medicare and Medicaid payments combined. This cost expected to rise to achieve a high level of $1.1 trillion by 2050. The impact on healthcare system, people with AD and other dementias have more than 3 times as many hospital stays as other older people. And I do not know if you have this information that there is certain village in Germany and Belgium that these small villages are inhabited by patients with Alzheimer's disease. So nowadays there is a community for patients with Alzheimer's disease and can have an extra care in these villages. So they have more than 3 times as many hospital stays as other older people. Emergency department visits, home health care, and nursing home stays are more frequent for individuals with Alzheimer's disease. The last one, the impact of Alzheimer's disease is on the quality of life. Of course, the severity of Alzheimer's disease will impact quality of life. And it is a very important disorder that we could expect decrease in the social economical status of the individual if he experienced Alzheimer's disease. Of course, when we are in the 3rd stage, for example, of Alzheimer's disease, the patient is unable to have a continuous work, to go to work, and this will impact the daily life activity of these people. And so there is an important and a serious impact on the social economical status of the individual. This decrease in the quality of life will result to progressive cognitive decline, loss of independence, and increased dependence on caregivers. And you should know that we should equip and give extra information to the caregivers, because sometimes some caregivers, and especially the relatives of patients with Alzheimer's disease, suffer from depression, because they need extra care and they need to talk for a long time with them. So they need special care for the caregivers. The psychological impacts on caregivers include increased rates of depression and anxiety. We are focused now for the drug repurposing and repositioning for Alzheimer's disease. I read a recent article that mentioned that there is a certain survey for repurposing of drugs for people suffering from Alzheimer. And I see recently a TV show, they asked some patients with Alzheimer or early dementia in certain nursing homes, what are their opinion about drug repurposing, and if they agree to be included in a certain survey on whatever they accept the concept of drug repurposing or not. And we find that most of them are willing to join this survey, because they told us simply, there is no problem to join, and if there is benefits, it's okay. And I do not think that there is a risk to try another drugs which are FDA approved and EMA approved, of course, for another conditions to be used for management of Alzheimer's disease. So the concept of drug repurposing involves seeking new medications or molecular targets different from a drug's known activity, pharmacological effects, or binding specificities. Given the increasing rate of drug failures in clinical trials, drug repositioning has emerged as a promising strategy to mitigate the risk of such failures. You know that in the process of drug development, we have several phases and stages, which is very complicated and time-consuming and money demanding. So this process of drug development is very long process, and require much money. So by trying drug repurposing, we try to skip some of these phases in order to limit and decrease this time and money problems. So the benefits of drug repurposing is it is cost effective by reducing the financial burden of drug development, time saving that shortens the times needed to bring a drug to market, and of course it is a drug with a known safety profile. So the safety profile of repurposed drugs is well documented, reducing the risk of adverse effects, which is the main problem of the newly drugs which are introduced in our market. We have several stages and strategies of drug repurposing, and we start with identification of candidate drugs for drug repurposing. How I can identify potential drugs to be candidate for drug repurposing in general? Of course, it is by literature review at first, by thoroughly review scientific literature to identify drugs with potential for repurposing based on known mechanism of action. Then it could be through clinical data mining to analyze clinical trial databases for evidence of off-target effects or unexpected therapeutic benefits. And the last one is by high throughput screening using automated techniques to screen existing drugs against new biological targets or disease models. So these are the three arms of the identification of possible candidates for repurposing. The second one, of course, is by cooperating with other departments to do some in silico methods by computational biology using bioinformatic tools and databases to predict the potential new uses for existing drugs. The second one is by molecular docking to simulate how drugs interact with certain molecular targets. And we have in our labs using this molecular docking for phosphodiesterase enzymes. Network analysis to study disease drug networks to identify possible repurposing candidates. The third one is by experimental validation. And of course, we are in the middle stage between in vitro models and the clinical trial models. We are concerned with the in vivo models. So in vitro models to test the candidate drugs in cell cultures to observe any effects on specific biological pathway. And then we start our job on in vivo studies to conduct animal model studies to evaluate the therapeutic potential and the safety of repurposed drugs. And I will explain this in more details later. For clinical trials, we have phase 1, 2, and 3. Phase 1 to evaluate the safety, of course, and the tolerability and kinetics of the repurposed drugs in a small group of healthy volunteers or patients. And the second one, which is a phase 2, to assess efficacy and the possibility for side effects in a larger patient group. And finally, phase 3 tries to confirm effectiveness and to monitor the side effects, compare the repurposed drugs to standard treatments in a large-scale ones. After that, we almost finish our job, and we will start the regulatory consideration by intellectual property through assessing patient issues and consider filing new patents for repurposed indication, and the regulatory approval, of course, by FDA and EMA. The last stage is commercialization, the market analysis to conduct the market research to understand the potential demand and the competitive landscape of this repurposed drug, and to try to find partnership to collaborate with pharmaceutical companies and biotech firms, as well as academic institutions for development and the marketing. And finally, the job of pharmacovigilance or post-marketing surveillance to monitor the long-term safety and efficacy of these repurposed drugs once they are released in the market. This is a very important diagram showing the drug repurposing in Alzheimer. You find here that the process of drug repurposing in Alzheimer started in 2012. We find that the publication per year is 1 in 2013. And when we say that there is an increasing number of publication of repurposing drugs in Alzheimer's disease, and for sure you notice that the largest one, or the high number of publication, were found in 2021. Why in 2021? The COVID, there is the COVID surge, and there is trying to find the drugs repurposed. And as you remember that in Alzheimer, in COVID-19, there is many drugs which are repurposed for COVID-19, like anti-malarial drugs and other classes. And of course, for this reason that we found that the highest number of publications were found in 2021. And then if you notice that it comes down again in 2022, it is decreased. So out of 53 clinical trials listed on clinicaltrials.gov, a total of 58 repurposed drugs have been reported. These trials include 15 agents in 12 phase 3 trials, and 33 agents in 32 phase 2 trials, and 10 agents in 9 phase 1 trials. Additionally, 6 agents are involved in phase 2 and 3 trials, and 2 agents are part of phase 1-2 trials. Some trial studies more than 1 agent, and certain drugs are being investigated in multiple trials. This is just a summary of repurposed drugs in general. We find that these drugs are unique repurposed agents represented in the current Alzheimer disease. We find that there is a large number of hematological and oncological agents, 18% for cardiovascular agents, including all classes of antihypertensive agents, and psychiatric drugs for 14%, 12% for antipsychotic drugs, I think, and 16% are multi-drugs for different purposes. So there is 15 unique repurposed agents, which are very promising to be repurposed in Alzheimer's disease. What are the current repurposed drugs in Alzheimer's disease? The first one in many, not only in Alzheimer's disease, but also in different disorders, is a metformin. It is a gold standard for repurposed in many disorders. It is originally used as for management of type 2 diabetes, and concerning its potential effect in Alzheimer's disease, it was found that it improves cognitive function by reduction of amyloid beta burden and enhancing neurogenesis and modulating insulin signaling pathways in the brain. The second one is the repurposed antihypertensive drugs, and I think that the first drug that we tested in our lab is perindopril, which is an ACE inhibitor, and it is one promising drug that crosses the blood-brain barrier, and we found that he has a great impact in management of brain inflammation and improving the cerebral blood flow, and potentially decrease the cognitive decline which occur during Alzheimer's disease. The third class is antidepressant drugs like citalopram and fluoxetine. Their main effect is antidepressants, and their beneficial effects for Alzheimer's disease is that they reduce the amyloidogenesis and alleviate some neuropsychiatric symptoms associated with Alzheimer. As you know, that Alzheimer's disease in the later stages, the patient does not suffer only from Alzheimer, but there is an anxiety, depression, and many other psychological disorders. So we find that these antidepressants could achieve many effects, including inhibition of amyloidogenesis, together improving the neuropsychiatric symptoms. We have, of course, the anti-inflammatory drugs, because it was proven that inflammation is a very important hallmark also of Alzheimer's disease. And it is included in the theories of Alzheimer. And as I mentioned with many professors here, that we do not know precisely who comes first, is the inflammation or amyloidogenesis. Whether the inflammation is the start of the process, and then it will trigger amyloidogenesis and topathy, or the amyloidogenesis and deposition of the neurofibrillary tangles are the reason for the inflammation or the inflammatory status in the brain. So by using some anti-inflammatory drugs, we found that the rofecoxib and the etanercept, when they are repurposed which are usually used for management of many inflammatory conditions like inflammatory bowel disease and rheumatoid arthritis or osteoarthritis, it can also have an impact on Alzheimer's disease by reduction of neuroinflammation, of course, which is a key feature in Alzheimer's disease pathology. We have another group of antimicrobial agents like doxycycline and rifampicin. They originally used as antibacterial, and they are found in many manuscripts that they reduce the amyloid burden and they mitigate inflammation, contributing to neuroprotection. We have another one, which are the anti-cancer drugs, like the sutinib, which is used for management of many types of cancer as immunomodulators. It will inhibit pathways that lead to neurodegeneration like angiogenesis and abnormal cell growth in the brain. We have certain immunosuppressants like sirolimus and fingolimod, which are used usually for multiple sclerosis and organ transplantation. These drugs may reduce neuroinflammation and enhance the neuronal survival. As you know that the most important impact of Alzheimer's disease is neurodegeneration or neuronal loss or neuronal death. The last one is anti-diabetic drugs. Of course, we mentioned metformin at first, but this drug is another category. Other drugs like liraglutide and pioglitazone, they are used also for type 2 diabetes, and they enhance insulin sensitivity, and they have neuroprotective effect through reduction of oxidative stress and inflammation in the brain. We have also anti-epileptic drugs and anti-hypercholesterolemics. Both they stabilize the neuronal activity and reduce the excitotoxicity and the proven anti-inflammatory activity. So in short, to summarize this, the drugs used as repurposed drugs for Alzheimer's disease, they will improve the symptoms through their anti-amyloidogenic activity, antioxidant effect, decreasing the excitotoxicity and release of the excitatory neurotransmitter like glutamate in the brain, and of course as to manage neurofibrillary tangles, and to exert also anticholinergic effects. As you know, that acetylcholine is a very important neurotransmitter for memory. By increasing the level of cholinergic neurotransmission in the brain, we can achieve a great impact on the symptoms of Alzheimer's disease. Now the last part in my presentation is what we are doing regarding the repurposed drugs in Alzheimer's disease in our lab. In short, as a class of anti-diabetic drugs, we have allogliptin, which is dipeptidyl peptidase 4 inhibitors. We tried to see the effectiveness of allogliptin by reduction of lipopolysaccharide-induced neuroinflammation in mice through targeting modulation of toll-like receptor 4, MYD88, and NF-kappa B, and of course the microRNA 155 SOX1 signaling pathway. Usually when we start working in our lab, we start by selecting the pathway. Then we see the impact of the drugs as, for example, here as anti-inflammatory effect, and then we select the model. Here we selected the model of lipopolysaccharide. This lipopolysaccharide, which is the LPS, we tried in this manuscript to inject it intracerebroventricular in the brain of mice. Okay, we use I think 1 milligram per kilogram of lipopolysaccharide, and then we injected it in the brain, and after 7 days by histopathological examination and the behavioral examination, we find that it is the symptoms of neuroinflammation are present in the brain. So after that, after 7 days, we start our work by providing our students to do some behavioral test and some biochemical investigation of biomarkers, and then histopathology and immunohistochemistry. The second one is baricitinib. Baricitinib, it is used in the market to manage rheumatoid arthritis. And in our recent, it is a paper published in 2024 by repositioning of baricitinib for management of memory impairment in overoptimized degalactose-treated rats. And we searched the signaling pathway of the potential role of JAK2, STAT3, PI3 ACT, mTOR signaling pathway. Here in this model, it is a different model where we used overoptimized mice. As you know, there is a great impact of estrogen in memory. So we expect usually that after menopause, we suffer from some sort of memory loss or decrease. We tried to see the impact of this overoptimized degalactose mouse model for management of memory impairment. We are here concerned with the memory impairment and the impact of this signaling pathway in this model. The third one is a formoterol. Formoterol, it is a beta-2 agonist. It is used usually for asthmatic patients. And it is one of the most important papers that we applied, I think, in 2018. And we target here the central beta-2 receptors that ameliorates streptozotocin-induced neuroinflammation via inhibition of glycogen synthase kinase 3 pathway in mice. Here we are focused on the insulin resistance in the brain. So our model to induce a status of insulin resistance in the brain is by injection of streptozotocin. Streptozotocin is a compound that used previously many years ago to induce diabetes type 2 in mice and rats. But when we searched for more details about streptozotocin, we found that there are trials to use it in very much lower doses. For example, in the induction of diabetes, it is given intraperitoneally in mice and rats, and it is given in high dose of about 60 to 200 milligram. But in Alzheimer's disease, we try to inject it intracerebroventricularly in very minimal dose, 1 milligram up to 3 milligram per kilogram. It induces status of insulin resistance in the brain, and we find that it induces what is known as Alzheimer type 3. Okay, diabetes type 3. Okay. After that, scoparone, it is a phytochemical compound. Here in this manuscript, we cooperated with another lab in the Medical University of Lublin that they worked on scoparone for many psychological disorders. And we tried to see what is its impact in also ovarectomy, degalactose, Alzheimer's disease. But in this paper, we are focused with the NLRP3 inflammasome inhibition. The fifth one is another phytochemical compounds, which is a mangosteen xanthone compound. We tried to see What is its impact? Because this compound has a great anti-inflammatory effects that are known previously, and it has some impacts on the insulin signaling pathway. And for this reason, that we try data in the streptozotocin model of neuroinflammation. This is similar to the previous one, which is a eumohenegrine. And we have tadalafil and bergaptan. That tadalafil, you know that it is a phosphodiesterase inhibitors. We try to see their impact in also the streptozotocin induced sporadic Alzheimer's disease. And by the way, all our work is on sporadic Alzheimer's disease. You know that Alzheimer's disease, there is two types. There is the sporadic one, which accounts for most of more than 95% of the reported cases of Alzheimer. And we have another one, which is a familial one or with certain genetic predisposition. The last one, I think that it is a dansotoxin and amblyfrone. Also, this is one of our recent manuscripts, which is focused also on JAK2, STAT3, NERV2, hemoxygenase 1 antioxidant pathway, which is focused on also the anti-inflammatory and the cognitive dysfunction effect of Alzheimer's disease. Thank you, and I hope you enjoyed it. Thank you.
The question? Sorry. Okay, have the question for no question? Professor Sancini?
Thank you for your talk. My question is related to any kind of repurposing related to lung-oriented or lung-targeted drugs? No?
No, you mean that for the lungs?
Sure. You know that inflammation will start also from lung effect. And my concern is that is some relationship between disease starting from lung dysfunction could end in spreading of the inflammation over the brain due to the lung-brain axis. And my question was related to any tentative to find out any.
Yes, I will try to search.
Okay.
And I will try to implement in our lab. But once I get about this, what I think that between the lungs, as you know, that there is a link between every parts of the body.
Sure. Gut and brain.
And we found that there is a big impact between this axis and the brain. So it is very interesting.
Oh, we can talk later if you want to the lung-brain axis. Thanks.
Any question?
Thank you so much. I have just a curiosity concerning the patenting of these kind of drugs. Do you think that is more easy to patenting because the drug is the same but with a new function, or there are some tricky or concerns about the patent?
But on the national, yes, we have, I think, 3 patents about this repurposing of these drugs, but it is done on the level of Egypt, not worldwide. But we have about 3 patents concerning these repurposed drugs, with of course recommendations. We try usually to make certain recommendations for clinicians and if they want to prescribe, for example, certain antihypertensive drugs or antidiabetic drugs, we shift them to use this. Yeah, of course, of course.
What about concentration of these drugs? Are they similar to what we have already used to treat other pathology targets, or are higher, and what higher?
In most of our work, we usually work on different dose levels at first. The one which is approved in the market, and one with a lower dose and higher dose. And at first, we perform certain pilot studies at first, and we use 3 different concentrations. And then we apply, at first, we apply certain behavioral testing and histopathological testing. And then according to these 2 tools, we conclude if the one which is present in the market, which is used, or lower level or higher level.
Thank you.
Another? Okay, thank you, Nesrine. Oh, the last one.
Thank you for your talk. I was curious about the possibility to use this repurposed drug in therapy of Alzheimer's disease together with the currently used drugs, and which is according to you, the best timing to give these drugs to the patients?
Of course, the best time, I will start by this point, the best time is in the early stage or middle stage, of course. But I think that even if the approved drugs now or these repurposed drugs, if they are given in later stages, the percent of improvement is lower. And I think that we discussed this in the morning. So I think that this is most important to implement early diagnosis and to use these drugs in early stages. Concerning using with approved drugs, I think that most of the approved drugs now are monoclonal antibodies. And of course, there is some drugs like anticholinesterase inhibitors, like donepezil, like other drugs, memantine, but all of them, they are for symptomatic relief. Not they are used for to improve certain molecular signaling, to reduction of neuroinflammation. I think that, for example, donepezil is concerned with acetylcholine, which is already decreased in the brain. So I think that it could be done, but by reducing both doses. So I can, for example, we recommend metformin together with donepezil, and we can reduce the doses of both of them. We cannot give them in the full dose. Okay.
Very much. Student? Curiosity? No.
They are satisfied.
Yeah. Thank you,
Thank you.
Serena, for your seminar. Thank you very much.
Okay. Okay. Okay, the first one.
I believe that if I just, yes, I enjoyed it. It's a very clever approach to reuse something that's already, we know it's safe, at least it should be safe.
Safety. It's about safety. Okay, thank you. Thank you very much.
This with the university. Thank you.
Thank you. Thank you.
Our
So,
close. Yes.
your job. Your
Yeah.
job.
Again, my job. What's this? Sappiamo la registrazione. Anna della Torre è stata.
Grande Anna della Torre.
Grazie Anna della Torre, se vuoi puoi chiudere il meeting.
Ok, grazie.
Sei stato, non so, capace di andare oltre.
Io